So that's my little analogy for genome assembly, but the actual scenario is almost exactly as I described. So we're going to start with short reads. In real reads, these would be like 100 base pairs long. We're going to scan through them, build up this De Bruin graph of how the different pieces of those reads are related to each other. So for example, this first read, AAGA, is going to be decomposed into these pair of nodes, AAG followed by AGA. We'll just keep doing that. That's going to form this initial graph. And then we want to be able to perform various simplifications on this graph to be able to spell out what the possible genome was. Notice there's two possible tours through this graph, one that goes through this top loop, then the bottom. There's another tour that goes through the bottom loop and then the top loop and then out. So that means in this particular graph, there's two possible genomes that could be spelled out. There's other information that we often have that I didn't describe that will help us decide which of these two reconstructions is the correct version to which of these two is the actual true genome. So the computation, is there's a nice algorithm for it, but it's very complicated just by the scale of it. So if we're trying to use this algorithm for the human genome, we're going to have many billions of nodes, many billions of edges that we need to be able to analyze and be able to process. There are a few algorithms that try to do this, but they all require either many terabytes of RAM, or require a cluster using MPI, or require a very big server with many hundreds of gigs of RAM to be able to compute to reconstruct the genome. The interesting part is just scanning through the reads and then building that initial De Bruin graph is actually quite straightforward to do in a MapReduce setting. It's actually just a pretty straightforward extension of that word counting example, where normally that canonical MapReduce example counts the number of occurrences of each word. Well, instead of counting the number of occurrences, you just keep track of which word comes next, and that'll build up the De Bruin graph in just like less than 100 lines of code. Really, the challenge is once we build this graph and then it's spread out over hundreds of different computers in our MapReduce cluster and our Hadoop cluster, how do we synchronize all this computation so that the different parts of the graph can be traversed, so they can be simplified, so we can recognize important motifs and so forth. So in particular, I want to take this graph where we have, this graph of just fixed length sequences, and then compress it down into this graph of longer segments. There's a paper that I helped write that describes the general scenario of how graphs can be efficiently processed in a MapReduce environment. But here we have this really special computation where we need to be able to traverse graphs in this very complicated way. So as a warm-up exercise, there's probably several hundred people on this conference. What if we wanted to compute who was born closest today? So who was born closest to July 30th? If everybody on this call tried to talk to each other at once, this would be really hard to do. So really what we want to do is, do this really localized computation where, you compare to your neighbor or maybe compare just to one other person at a time. So the key idea for making this work is we're going to organize everybody on this conference into some sort of bracket, into some sort of tournament setting where, you're going to compare to, say, the person sitting next to you. Then you'll find who was born closest today, maybe in your part of the room, and then maybe each of the different sites will figure out who was born closest today. And then finally, we can have some high-level communication where just the different sites will communicate with each other to figure out who is closest today. So in this case, we have a bracket tournament of 64 teams, and then in just 6 rounds of parallel computation, we can find the winner. In our case, we have a couple hundred people, so maybe we're talking 8 or 9 different rounds of computation. But through the power of log, if we scale this up to billions and billions of people, in just like 30 rounds of parallel computation, we can figure out who is born closest today. So there's this really nice scaling property. If we can organize the parallel computation in just the right way, it doesn't require very many rounds of parallel computation to solve even these very big problems. So in our case, we want to apply the same sort of idea to a graph problem. The problem is these nodes don't have any sense of, of where they are and who's connected to who. All we know, all that's known is this node knows it's connected to this second node. The second node knows it's connected to this third node, and it knows that it has an in-edge from this first node. So nodes only know about their local neighborhood. It doesn't know anything about the global structure. What we want to do is impose a bracket on top of this, so that we can have some sort of order to this. Now, trying to compute an exact bracket on top of this is actually a very hard problem to do in itself, but we can use a technique that goes back to the 80s that will allow us to apply a randomized bracket to all these different nodes so that we can structure the computation very, very efficiently. So the idea is for every one of these nodes, we're going to flip a coin. We're going to assign heads or tails to each node. And then every time we have a head node followed by a tail node, we're going to be able to then, in one round of parallel computation, execute that compression. So here I've assigned head and tail to every single node. Notice that can all be done in parallel. We can then identify pairs of nodes that it's heads followed by tails. This isn't going to identify every single possible pairing that could take place. But the good news is because it's randomized, we're going to get a very large fraction of the pairings that can take place. And then conceptually what's going to happen is all those places that are paired up in one round of parallel computation are going to be merged together. So we can simplify the graph like that. So in that case, we reduced the number of nodes by about 38%. We do this same sort of computation where we're going to pair up, where we're going to identify nodes and edges, we're going to identify nodes where it's heads followed by tails, and then we're just going to iteratively pair them up, merge them, compress them down. And then the good news is that in log of S, where S is the length of the longest chain, we're going to be able to maximally compress down this graph. So that's great because even if the longest chain is 1 billion nodes long, we're only going to need about 30 different rounds of parallel computation until the graph is then maximally compressed. That sort of graph compression is very fundamental to this genome assembly problem. There are other then sort of parallel graph algorithms that need to take place to recognize different motifs. So we need to identify something called tips, bubbles, dead ends, half branching, full branching nodes. In the interest of time, I can't explain how all those different parallel graph computations take place. But the remarkable thing is that it all works very, very efficiently in MapReduce. And then the system that we're developing called Contrail is going to start from billions and billions of short reads, in this case from a single human genome. The initial graph that we're going to have is going to consist of many billions of nodes and edges. We're going to do some of that initial compression to reduce it by about a factor of 10 into about 1 billion nodes, and then apply various error correction algorithms, various algorithms for resolving repeats, various algorithms for bringing in more of the data, and then instead of starting with billions and billions of nodes, we're going to shrink it down to about 3 million nodes, each of about 1,000 base pairs long. So this is a pretty good genome assembly for these data, taking billions and billions of isolated nodes and then reducing it to a few million with the different parts of the genome reconstructed. This same sort of assembly idea is also being applied in addition. So that was an example of trying to assemble the human genome. We're applying the same sort of idea to assemble multiple people at once. So in particular, I collaborate on this project where we're trying to study the genetics of what causes autism. So we can build up the same sort of graphs, but we can just color the graph by which member of a. So we're going to build up this same sort of graph, but we're going to build it for an autistic individual, and also that same person's parents in the same graph, and then color the nodes of the graph by who contributed the sequence to the graph. And then in that way, anytime we see that there's a set of nodes in the graph that were all contributed by that particular individual, we can say, aha, here's a mutation that is very specific to this one person. So at the bottom we have an example of a 6 base pair deletion that was deleted in a kid that had autism that wasn't present in that person's parent's genome, and was also not present in that person's sibling that was not autistic. So we think that this sort of mutation was specific to the kid with autism and may have contributed in a causal way to that disease.